Good afternoon. Thank you all for coming today. I want to welcome everybody to the Frontiers from Brain Health at the Center for Brain Health, where we take a deep dive into the most exciting new developments in brain research. I'm Dr. Jane Wigginton, an emergency medicine and lifestyle medicine physician, and I'm privileged to serve here at the Center for Brain Health as the Medical Science Research Director. I want to let you know that at the end, we'll have a few minutes for questions. So if you're viewing online, please use the Q&A feature if you have questions that you want to talk about. And if you're here in person, then go ahead and jot your questions down, and we'll take as many as we can at the end. This presentation is also being recorded for future viewing and sharing. For those of you who may not be familiar with the Center for Brain Health, it's a cognitive neuroscience research center at the University of Texas at Dallas, dedicated for over 3 decades to exploring neuroplasticity and the brain's amazing lifelong potential to get stronger and work better. So I want to now introduce Dr. Matt Huentelman, a professor of neurogenomics in the nonprofit Translational Genomics Research Center in Phoenix, Arizona. And he's the founder and the principal scientist of the MindCrowd Research Project. His research focuses on the molecular basis of human nervous system, health, and disease, aiming to personalize medical approaches to aging and optimize brain health for successful cognitive aging. This modern approach, known as precision aging, aims to align our cognitive health span with our physical lifespan, significantly reducing the burden of age-related brain diseases. And with that, I'm going to turn it over to Dr. Huentelman. Let's hear a round of applause for him.
Thanks so much. It's really great to be here. I appreciate everybody showing up today. My talk is going to be about 40 minutes long, so about 45 after the hour, we should have plenty of time for questions. So I really appreciate it. It's been really wonderful to be hosted here. I've really enjoyed my visit to Dallas, even though it might have been brief. It's been really incredible and very stimulating. So I'm going to tell you about 2 studies today. The first one being an internet-based study, and the second one being a mobile lab-based study. Before I start, just want to acknowledge my team. They do all the hard work. I get to come on the road and talk about their hard work. So that's what I'll do today. And the little star there, in case you didn't know where Phoenix, Arizona is, that's where TGen is, where our lab is based. And I also want to acknowledge collaborators, long-time and short-time collaborators, especially Lee Ryan and Betty Gliski from the University of Arizona, who helped create MindCrowd with me, as well as funding support, most importantly from the National Institutes of Health, but critically for MindCrowd, the support of Mike Mueller and the Mueller Family Charitable Trust. They really helped us launch this study. All right, so let's dive in. I know I have a diverse group here today, some hardcore scientists and some even community members, so we're going to try and be in the middle somewhere so we can all get on the same page and understand a little bit about what I'm talking about today. So first I want to go through a few concepts that I think really drive our research. You may disagree with these, but this is what drives me. First, I think that to understand the broken brain, so brains that are suffering from diseases like dementia, for example, we really have to better understand the healthy brain. And so we need to study more healthy people and follow them, see how they change across time, because that tells us what happens when the brain encounters disease. If anyone here in the room is a car mechanic, you don't usually start with a broken car. That's not where you end up with your passion for repairing cars. Usually you take a perfectly working car apart, and that's where you learn. And if you put it back together and it still works, then you've learned something. And so I do it too, but in science we tend to skip past the healthy part and we start to study the patients who have disease because we want to help out. But it's important to remember the healthy brain can tell us a lot. The other thing is we know how good the brain is at hiding problems. By the time Alzheimer's disease causes daily problems, We estimate that you might be missing about 70% of your brain cells in the middle of your brain in the hippocampus. And your brain's been hiding that from you as those brain cells die, because it takes around 10 to 20 years for Alzheimer's disease to progress to that particular spot in time. So we need to do a better job of preventing that disease. And one of the struggles is detecting it earlier, because the brain does such a good job of hiding it from us, from compensating. So by the time we see disease in the clinic, it's really hard to make a difference. So we need to focus a little bit earlier. And so along those lines, I ask you, when was your brain last tested? When you went in for your wellness visit, did your physician sit you down and give you a memory test? Very likely not. Very likely not. But did you get a cholesterol test? Absolutely. Did you get some preventative cancer screening? You probably did. So I admit it, we don't have a whole lot of medical interventions that can be helpful with the brain. No magic drug to prescribe, and maybe that's the reason why we don't test the brain. But I think we really need to change that approach. We all need more longitudinal assessments of our brain, and this needs to work its way into our wellness visits. Rapid fire topics for you guys today, just to get us on the same page. Okay, what is precision medicine? Because I want to focus a little bit on what I call precision aging today. And we've really learned about this from our friends who study cancer and oncologists in our life. This is a figure that's called the hallmarks of cancer. The field in general updates this figure as new things are learned. But you can see all these different pathways that lead to cancer. Biological processes that help the cancer grow, evade the immune system, resist medical treatments. Each one of these has been built over time through a lot of hard work in the world of cancer. And the really incredible thing is that for most of these biological pathways, they have medicines to treat it. And so we want to understand this exact same diagram, but for the aging brain. Now, we have a lot of work to do. We're many decades behind cancer, but I think cancer can tell us a lot about what to focus on, and we can follow in their footprints and build on those successes, especially what they've done in precision medicine. So precision medicine means use the cancer to tell you what drug to treat it with. Let the cancer unveil its Achilles heel, and then use a medicine that targets that, instead of just giving every breast cancer patient breast cancer medicine number 1, 2, and 3. Let the breast cancer tell you what drug should be used. So we formed this collection of scientists called the Precision Aging Network. You can visit our website to learn more, but the goal here is very simple. Apply that knowledge and that approach for precision medicine and cancer, but apply it to the healthy and typical aging brain. In other words, we all know what we should be doing for brain health. There's a long list. There are experts here in the room that can help you understand what can be helpful for your brain. But I think we all can also acknowledge that from this long list of things that are good for our brain, we don't have time every day to check off every one of those boxes. So I personally would like to focus on the most important things for my brain. And the top of that list might be very different than my friends. They might have different things that are important for their brain. So we're taking the very beginning steps of understanding what we should prioritize. And that's where you get precise and personalized in those recommendations for your brain health. Okay, so what can we learn from precision medicine and oncology? What did it teach us? There's 3 key things that happened in cancer biology which really drove this. Number 1, large, diverse data sets studying all types of cancers, all types of tumors, helped them understand what to focus on. We need the same thing in aging. A depth and a breadth of phenotyping, so really spending a lot of effort understanding what's changing and the characteristics was really, really important. And those 2 things are important because they help feed some of the advanced statistical models like AI and machine learning. And then lastly, we do have to test things to show that tailoring interventions leads to a better outcome than just a blanket suggestion about what to do about brain health. So we have a lot to do. I'm not going to solve these three things today. But I'm really excited about where we're moving as a field. Okay, so my work today was really born from an experiment that we did about 20 years ago. And what we did was we tested the memory of about 1,000 humans, and we did the genetics of that entire group of people, and we said what genetic drivers were associated with how they did on the memory test. And we found something, and at the time it was new and exciting and very cutting edge, so of course it made it into a great journal, Science. But that genetic foothold didn't really yield functional information, so we didn't understand functionally what was going on until really recently, where a group just recently published last year how this might potentially be functioning as it relates to human memory. So I'm just showing you this slide, number one, to say we've been grappling with this for a long time on my team. But number two, to say this study, which was, again, about 1,000 people, was really expensive and took a very long time. And it always bothered me that we wanted to do something larger, and I couldn't imagine scaling this up until we decided to have our study live on the internet, okay? And so that was the key to scaling this up. We started this about 12 or 13 years ago now. You're all welcome to join, mindcrowd.org. You can join us. Don't join right now. You won't be happy with your score. Or maybe you would if you ignore me. But I'll make it hard to ignore me. You can participate in English or Spanish. We're very proud of that. The whole experience is available in Spanish if you prefer to participate in that language. So let me tell you a bit about MindCrowd. But first, I want to start with a couple slides that help answer a few of your questions ahead of time. I am not proposing that we should end all in-person studies, okay? This is not a challenge of internet versus in-person research. They each have their pros and cons. In fact, at the end, I'll talk about how there can be an ecosystem that is really, really beneficial to have multiple aspects of your work that are both online and a mobile lab and in-person research. Secondly, many of you may have used Mechanical Turk from Amazon to do some of your piloting, to do some investigations in your own lab. That's not what this is, okay? Amazon mTurk, people are usually making money to participate in your studies, for example. It has its place, it's great for piloting things, but this is a true research cohort that's built de novo by inviting people from the public to join. And then lastly, I totally understand it. We're on the internet. There's a lot of questions about the data quality. And you do have to be very ruthless. You have to be very careful with which data points you accept as scientifically valid and which ones you remove. So we spend a ton of time. This is probably where we spend most of our effort, is cleaning the data and understanding what is a quality data point versus a non-quality data point. So that includes for things like removing people who are too fast. Believe it or not, it seems like we have bots that participate and they have faster than human reaction times, right? So it's really important to remove those data points. Sometimes people respond too frequently. So we see 25 responses when we only had 20 trials. So those folks are responding too frequently, and therefore they're typically removed from the research. So that's just a few examples. But we're very sensitive to this. That's all I'm trying to say. All right, so this has been incredibly successful. This is the MindCrowd cohort. We actually just passed 700,000 participants last morning. And each one of these little red dots is a participant from around the world. You can see we have some opportunities here. So we definitely need to launch a Mandarin version of MindCrowd. Maybe a Hindi version of MindCrowd would also be helpful. And we're actually working on those. But over 60 million data points from over 650,000 participants to date. So how do we do that? Well, we spend a lot of effort recruiting. We actually recruit about 700 new participants every day. So it's pretty amazing to give an hour-long talk to you guys and I'm doing science as I'm talking to you, which is really fun. Of course, the internet's open 24/7, so people can participate any moment of the day. And so just this last year, we recruited over 250,000 participants. And there's our plot across time there. Our stated goal at the start of this was to reach 1 million. And I'm pretty excited because that seems within reach now in about a couple years. Okay, one of the other powerful things about being on the internet is that you truly are open for business for the entire community, the entire population. And it's led to a really incredibly diverse research cohort. So over 84% of our participants in Mindcrowd are underrepresented in research studies. So this is a definition that was put forth by the All of Us program, and you can see the different categories below that it relates to. For example, educational attainment below a high school diploma. Those folks are very understudied and underrepresented. Various race ethnic groups, people over the age of 66, or 66 and older, I should say, are very underrepresented in science as well. But also participants from rural zip codes are also very underrepresented in our research. So one of the powers of having your study live on the internet is that you really can reach a very nice, diverse cohort, which is really important. Think back to what I mentioned about the diversity being important for some of those original precision medicine trials. This is helping us address that. Okay, so how do we do it? Well, we very active social media programs. We're very present on Facebook, X, and Instagram. But one of the new things we did was to utilize this resource called Tapjoy. So how many people play games on their smartphone? Yeah, none of you are telling the truth. Okay, y'all have games on your smartphone, I know you do. A lot of people do in society. It's very frequent to use your smartphone for gaming, right? Inevitably, you run out of that free money in those games and you need to buy more diamonds, right? So the way you buy more diamonds is you use real money, or you can do stuff and get rewarded. So these are called offer walls. So you might find that joining Mindcrowd could be on one of these offer walls. So if you join our study, you'll get 10,000 diamonds and you can continue playing your game. This approach is incredibly powerful. So look at what it did to the diversity here. Dramatically improved the high school education or less group, dramatically improved the underserved, underrepresented race groups, and I'll show you a visualization of this. The numbers are kind of hard to read, I'm sure. Was a real incredible improvement. So we've been doing this here for the last 8 months. And here are the results for that. So you can see it took, sort of in the upper left there, it took our white percentage in Mindcrowd, white race, from around 78%. It's now down to really where I would want it to be, which is closer to 60%. And you can see Black African American participation went from around 5% now up to around 12 or 14%. So that's just one example of some of the real benefits of this recruitment approach. I'm just telling you one vignette here, okay? There's a lot of other approaches we take that you can take as well. But my point is, you always have to be recruiting. You have to be thinking about this and changing your approaches. But the other powerful thing is, some of the approaches you choose can change the demographics of the cohort that you recruit. So a lot of people fall into this trap of, okay, the cohort I'm recruiting is going to stay the same forever. You can actually tweak that by being creative. So really interesting. The other place that most people imagine is that an internet-based cohort must be full of people who have lots of money. Don't understand why that's an idea. The internet is pretty affordable for most of us to get on and is used across all societal groups. And so I'm just showing you here the socioeconomic status within Mindcrowd. This is just a snapshot But around 15% of Mindcrowd participants who are 30 years and older, so that's an important cutoff to kind of remove some of the typical student ranges, have a household income below $35,000. So we do see a very nice range of SES in our cohort as well. All right, so let's look at some of the demographics together. So these are numbers from Mindcrowd from the past 12 months, okay? So just looking at what we've done over the past year. And I'm showing you education, sex, ethnicity, race, and the ages here for Mindcrowd. And so what you can see is that while we do have some high education in the cohort, the high school diploma and less group is close to about 40% of Mindcrowd. So that's something that in science we would say is a low education group. That's very powerful. 2/3 women, 1/3 men, very typical for research studies. Those of you who do research, women are much more altruistic in giving up their time. Even though we're only asking for 10 minutes, that still holds true. Hispanic, Latino percentages around 22%. So that's a really strong place for us within Mindcrowd. And you can see the race breakdown as well there with, again, over the past 12 months, about 62% white. Age decades. So again, a lot of people think, oh, this is an internet cohort. It's full of people who are 30 and younger. Absolutely not true. You can see our #1 recruited group is between the ages of 60 and 69. So 22% of our participants are right there. And also I can draw your attention here to the 80 and older. 80 and older group, 4.5% of our cohort is 80 and older. So you can look at the ends there, which is really quite striking. Every time I look at it, I'm still surprised. That's like 9,000 people over the past 12 months recruited from that age group. All right, deep breath. You made it through the first part. Okay. So here's the Mindcrowd site map. I'll give you 30 seconds to memorize it, and we'll do a test. Now, all I'm trying to show you here is that it's pretty complicated, and we're only going to talk about sort of the bright purple boxes today, which is like the main Mindcrowd experience. We have a lot of other experiences that I'm happy to chat more about, but this is the 10-minute experience. And I'm only going to talk to you about one of our cognitive tests. It's this test, the Paired Associate Learning Test. I love this test because it was developed over 100 years ago. I'm not trying to convince you guys that we invented something new. We just reduced it to practice on the internet. So I love this because we really know quite well how this test works, what brain regions it utilizes. That includes the medial temporal lobe. And Alzheimer's disease is one of the diseases that are most sensitive to the MTL, and this test is really quite hard for someone who has dementia. So these are word pairs that are not semantically related. However, we do have 3 word pairs that are. So 3 easy word pairs and 9 difficult word pairs. We show it across 3 rounds, so you get 12 words. You see them as 2 seconds each, so the word pair shows up for 2 seconds. You're asked to memorize it. And then you have to recall it immediately, and that whole process repeats 3 times. The beautiful thing is, most people learn on this task. We can see people's scores improving as they see the word pairs for the 2nd round and the 3rd round. But let's look at the data. Oh, sorry. That's Mary Whitney Calkins, who really invented this, one of the first women in the field of psychology in 1894. So pretty awesome. All right, here's the data. It looks quite crazy, right? I mean, this is a whole lot of data points. So this is almost 700,000 data points. Along the x-axis is age, from 18 years up to 95. We cut it off at 95 because that's the oldest person who I've actually spoken to who joined Mindcrowd, okay? We have a few 110-year-olds. I don't think they are 110, but I've never talked to them yet. So we just cut it off there, okay? A couple things, right? The presumption would have been that 18 and 20-year-olds would just be so good on this test. They would just be outperforming anyone in their 50s, 60s, 70s, and 80s, but that's not what we see. You see this incredible spread in the variation of the data. The trend is shown there in the line, so very clearly there is a trend to do worse on this test with age, starting at about an average of 20 and decreasing to about an average of 10. A perfect score is 36, okay? Just to remind you, 12 word pairs, 3 rounds. But I hope you can appreciate that amazing diversity in performance. And in fact, you can see some people in the 60s, 70s, and 80s who are performing really quite well, as if they were the average 20-year-old, or even better. That's fascinating to us. Really wasn't known until we did a large study like this to see that whole collection of data at once. Let's talk about a few of the trends for you guys. First, biological sex. So in pinkish are the women and blue are the men. And so at every single age, women are better at this task compared to men. The confidence intervals are in there, by the way. You just can't see them on the shading. They're so tiny. This was known before, but never under one roof. This was never shown in using a single task, a single test, covering the entire aging spectrum. So even 18-year-old women are better at this test on average than 18-year-old men, which is really, really quite impressive. And you can see that difference in the sexes just spreads with age. And here it is as a curvy line. And so I show this because we're very intrigued by this bump here in the women in pink. That happens right around the average age of menopause onset. So a lot of women come up to me afterwards and they say, no way, man, my memory got a lot worse during menopause. But the data that we have here suggests that that's not the case. On average, women tend to improve on the performance on this task, starting around the age of menopause onset. So a really interesting thing that we see that we're still investigating. Okay, let's look at a few other things. How about the response time? So how quick are you at answering these questions? All right? So again, we're looking at men and women in this case. The women are the red line, the men are the blue line. And on the left side, we've got correct answers. On the right side, we have incorrect answers. And what you see is that the men are quicker with their answers, both for the correct answers and the incorrect answers. So when they're getting them right, they're faster than the women at putting in those correct answers. But you can see with age, there are some changes here, right? So with the correct answers, the men tend to get faster and faster across age compared to the women. With the incorrect answers, the two sexes get closer and closer across age. So, I don't know, I guess both men and women have something to take home as a badge of pride, perhaps, today. This is one of our original findings, which we continue to replicate as our study grows, but it's really quite interesting. This is the influence of your family history of Alzheimer's disease on your performance. And on the left side, I'm showing first-degree relative family history, and on the right side, it's second-degree relative family history. First-degree relative means mom, dad, or siblings, and second-degree is the next level up of your family tree. So the reason why we split this up is because the first-degree relatives tend to have much more relationships when it comes to genetics and environment. And you can see the lines are only different on the first-degree relative side. And it's interesting because they're even significantly different in the youngest ages here, in the 18-year-olds, the 20-year-olds, and 30-year-olds. What this is saying is that if you have a family history of Alzheimer's disease, a first-degree family history, you actually perform worse on this memory test than your peers who do not have a family history of Alzheimer's disease. These are not college kids who have dementia, okay? But they are performing worse on this test by about 10%. So if you really dig into it, really test them, and if you have the numbers that we have, you can see that whatever's captured by this first-degree family history variable is associated with performance, which is really, really quite striking. Another one I wanted to show you was this cardiovascular risk factor effect. And this is really beautiful because it's dose related. So what we did was simply score the entire cohort for their cardiovascular disease or risk factors. That included diabetes, heart disease, stroke, smoking status, and hypertension. Okay, so there were 5 things, and all we simply did was add them up for the composite. So if you said, I'm a smoker, but none of the other ones apply to me, then you got a score of 1, etc. Okay? And then I'm just showing you data for the 50-year-olds and older, and you can see a very nice stepwise effect of cardiovascular risk factors. So the more risk factors you have for cardiovascular disease, the worse on average you perform on this memory test. So it's really quite beautiful to see really how it's almost stepwise in its effect. Okay, I wanted to dig into a few things about how we address some of the data quality and also self-report errors. So obviously, we're not interviewing these people face-to-face. We're asking them, do you consider yourself a smoker? And then they have to say yes or no, right? And so a lot of people like to wonder about that. How accurate is it for people to endorse things like that on the internet? So here's the main finding for smoking, and let me show you how we address this. So on the left side of this plot, we're comparing 2 groups, which is people who declared they were smokers or not. And in this case, I apologize here that I have the colors. No, the colors are correct, sorry. So the non-smokers are in gold, and the smokers are in blue. On the left side is women, on the right side it's men. So what you see very clearly is that the 2 lines are only different in the women. So smoking only has an effect in the women in our study. And it's a negative effect. So if you're a smoker, you perform worse on this memory test. Now guys, this is not men in the audience. Let's talk on the side here, okay? You don't get to go out and now pick up smoking. We need to protect all of our function on this test as we can as men, okay? But this is the finding. Let me walk you through this, because some people said, okay, what if folks are not being honest about their smoking status? That's a valid concern. It happens with all of our research, all of our human research. The thing that bothers me a bit is it also happens in the face-to-face studies, right? How do we know people face-to-face are telling us the truth? You know, I think there's pros and cons to asking some questions on the internet. And in fact, there's research that backs that up. There's research that has shown certain sensitive topics are answered more truthfully when you're not face-to-face with a human. Think about things like drug use, abuse, other sensitive things like that. And maybe smoking is one of those, I don't know, but let's assume that not everyone's telling the truth. I think that's a safe assumption for all of our human research. Here's what we're able to do. We just introduce additional self-report error. So we actually switch people's actual answers, and then we redo the scientific analysis, and we do that 10,000 times, and then we do it again. We introduce more error. And so that's what this plot shows. On the far left is the effect that we found, and then each one of the boxes has more and more self-report error. In other words, people were lying about their smoking status. And we introduced that artificially. And what the boxes are showing you, the box plots are showing you the distribution of the p-values. So is this a significant finding or not? The take-home message is pretty simple. We can introduce up to 7% for the main effect, 7% additional self-report error before even one single of those 10,000 studies ends up being not significant, okay? So, you know, I don't know what the self-report error is. No one really knows what it is here, but what I can tell you is if you assume there's some in there already, we can add more and more on top of it, and our effect remains significant until we reach levels of above 7% additional error. Now this is different for each finding, and we explore this for each finding. How much self-report error would basically make our finding go away? So it's a statistical way to start to investigate the role of self-report error in the strength of your finding. Okay. I'm actually going to skip this particular part because I want to talk about genetics. Actually, I want to emphasize this. We love to help other scientists. It's one of the reasons why I'm here today, talk with the great team here and talk about how we can work together. And we've already been doing that. So facilitating other scientists to use our cognitive testing platform, but also helping recruit into their studies as well. So these are just examples of some studies that we facilitated. So we're happy to collaborate, if anything, got you excited today about what I've said. All right, so we do collect genetics on our group. We do this remotely, so we don't see anyone face to face. They prick their finger and they send us dried blood in the mail for us to analyze. About 5% of people participate. And when we get their samples, we do what's called genome-wide genotyping. So this looks at about 1 million different genetic variants in the human genome. And we can do two important things with that information. Number one, we can look at this gene, which is ApoE, especially the epsilon 4 variant of ApoE, which is associated with higher Alzheimer's disease risk. And what we can show is that in a dose-dependent fashion, ApoE4 allele is associated with a change in performance. So that's shown in the box plots and the line plots. It's not that dramatic, but it definitely does have a significant effect. Recently, there was a proposal that people who had 2 copies of the E4 allele should just be considered they will progress to Alzheimer's disease, they will have Alzheimer's disease. There's no way to avoid it. And so, of course, that doesn't sit really well with me, nor with many other people in the field. And what I'm showing you here on the non-gray portion, I'm having you focus on those red dots. Those are E4 homozygotes. They carry 2 copies of the E4 allele. They're over the age of 65, and you can see plenty of those red dots are actually above the average line, meaning they're performing better than we would predict compared to their peers. So I think it's really dangerous to tell those folks that, hey, we think you might have Alzheimer's disease dementia, or you will have it and there's no way to avoid it. They're performing really quite well on this task. So the last thing we can do is calculate what are called polygenic risk scores. So these are things where we can use existing data to calculate your personal risk based on your genetics for all different types of human traits and diseases. We've done that here as well. The take-home message is there is no significant predictor of performance from these polygenic scores. However, I'm showing you sort of the cloud over there, right? Not very impressive. But that is considered to be one of our top predictors of performance, and it ends up being your genetic risk or your genetic score for intelligence, okay? Intelligence is very murky, but there's been large studies done on IQ scores, and that is our top predictor of your performance on this task. However, I will say after we correct for all the multiple hypotheses, it's actually not significant, which isn't too surprising because I'm showing you how it looks visually, right? You wouldn't be too compelled by that. You wouldn't run home and tell your family what you found in the lab today. All right. Good? So, okay. That was Mindcrowd, and I'm going to do 5 minutes on the mobile lab study, okay? So, this is the Mindcrowd cohort in the United States. The blue dots are rural participants. We have about 10% rural participation, really high participation rate, one of the other benefits of being on the internet. And those folks are extremely understudied, and they're at much higher risk for diseases, and they have worse outcomes as well. So I'm showing you here basically the top 10 diseases that kill Americans, and all the darker bars, which are rural, they're at higher levels. Okay? Again, we're in a very unique place because we live on the internet, so we see 10.9% here in Mindcrowd. So we wanted to go visit them. So we took a horse trailer and we wrapped it with our branding, and we hooked it up to an F-250 and wrapped that in some cool branding as well, and sent this on the road. So that's a picture of the truck and the trailer in rural Arizona in this city called Wickenburg, about an hour from Phoenix. So it's only 20 feet of flat space, very drivable. You don't need a CDL. But the key thing is, in the back of it, we stuck this ultra-low field MRI instrument. So this is pretty cool, 0.06 Tesla. It's the hyperfine swoop. It plugs into the wall outlet. It does not use liquid helium. And the fringe field is very, very small. It's the size of that hula hoop on the top of the instrument. So it's a very safe instrument to operate. It weighs about as much as a car or a horse. So you can put it in the back of the horse trailer and drive it around. So we've published a couple different important papers that showed how well it performs compared to higher field MRI. And we also discussed how to build this trailer and actually published it. That's one of my favorite publications because one of the co-authors is a mechanic. So that's pretty cool. That should be a goal of everyone, to get a mechanic to be a co-author on the paper. Okay, so does it take good pictures? No, it takes bad pictures. Okay, I told you, it's an ultra-low field MRI. Are you listening? 0.06 Tesla. But we live in the age of artificial intelligence. So through AI, you can morph these pictures into pictures that are equivalent to 3T or 1.5T images. The correlation between brain volumes is 0.999 with those. So the same person imaged in the ultra-low field and the high field, the brain volume calculations are extremely well correlated. So here's our mobile lab visit that we conduct. It's about 90 minutes. They do a typical survey that you guys probably all do with your participants, learn more about them. We measure their blood pressure, their grip strength. We have a smell test. We have them complete the 2 Minecraft tests, the one that I showed you, and a different one that looks at processing speed that we didn't talk about. We measure their body composition using one of those fancy scales that uses electrical impedance. We collect blood from their shoulder using a small capillary collection device. Then we run the MRI, which takes about 40 minutes. We get T1, T2, and flare images. And then at the very end, we take a picture of their retina. So we get a retinal image as well. So lots of phenotyping. Again, think back to what I said was what we learned from oncology. We need deep phenotyping to really power this precision aging approach. So that's what we're doing here. The demographics track extremely well with rural Arizona. About half of our cohort reside in rural zip codes. So we do collect data from people who live in Phoenix, for example, but half of it is from rural zip codes. They follow the demographics of rural Arizona, perhaps higher education levels in this particular cohort. But look at the age range. I really am amazed by this. So between the 20s and the 70s, we're averaging right around 15% of the cohort comes from those age groups. Those are groups that are really hard to recruit for a face-to-face study. When you bring the lab to them, they do come out and participate. Okay, Minecraft results. So I'm showing you that this cohort, so now this is just the mobile lab cohort, follows the same type of change in memory across time. And then the brain imaging results look very similar to what's been published. I'm showing you 4 different areas of the brain that are measured here: the gray matter, the cerebral white matter, the subcortical gray matter, and the ventricular volume. All these follow trends that have been published before, so I'm really happy with how the ultra-low field is working for these known trends. And I'll show you 2 more bits, and I'll finish pretty close to when I promised. Here's how the hippocampal volume changes with age. So the hippocampus is in the middle of your brain, and it's really important for performance on the memory test that we talked about, okay? So the hippocampus, you can see, is pretty stable in size between the ages of 20 and 40, but then it starts to decrease with age. So it starts to shrink in size. And this is corrected for the size of their head and the intracerebral volume. Interestingly, in people who are 60 and older, there is a correlation between your hippocampal size and how you perform on the memory test. So that's what I'm showing you here. Across the bottom is performance on the test, and on the y-axis is the hippocampal volume size, showing that general trend of correlating with performance. And the last thing I want to leave you with are the retinal scan results. So this is really fascinating to me. The retinal scans tell us a lot about your cerebral vasculature. The retinal vasculature is a good proxy for your cerebral vasculature. Helps us get some idea about perfusion and blood-brain barrier function. And we're already seeing really nice associations that others have shown with fractal dimension, a measurement of your retina correlated with age. But we're also seeing changes in your retina that correlate with how you do on the memory test as well. So I'm really excited to be collecting those data as well. All right, last thing is to take you back to this slide and just emphasize that there's real power here in combining a face-to-face study with an internet-based study with a mobile laboratory study that I described. Neither one of those three are perfect in isolation, but together the pros and cons really add up to a really powerful way to do human research. So what I want you to do today is to leave here and really start to dream about how your study might exist on the internet, or how a portion of it might. Maybe you recruit through the internet, but you see them face to face. It really does open up new ways of reaching out and really expanding your work a little bit. And I'm happy to take questions. Thanks, guys.
Thank you so much for that fascinating talk. It was so wonderful and so informative, and we're just looking forward to collaborating with you on some of these things.
Yes, same here.
Does anyone in this room have any questions for Dr. Huentelman? Okay, Steve?
I'm listening, Steve, but I'm going to drink something at
Good.
the same time.
Good. It was nice to see that picture of Four Peaks in the background for those who haven't been to Phoenix. That was Four Peaks in the sunset.
Yes.
You talk about risk factors, and when we talk about brain health, part of our belief is treat it like you would, when should you plant a tree? Start treating your brain better today. So instead of risk factors, is there some thought given to maybe calling these performance enhancement factors, right? Because when you're talking to certain groups of people, they don't want to know the risk. My mom passed from Alzheimer's, and I didn't want to know until I came here and learned more about it. Can we talk about both sides of that equation, and what have you learned on that score?
Yeah, I think that's a wonderful point. I think that sometimes the language we use in academic science doesn't translate well to the people who we're trying to help. I think that's your general point, which I think is a really important one. So absolutely, we need to do better with how we talk about these, because it's not all risk. Some of it's about resilience. Some of it's about protection. Some of it's about performance. And so absolutely, without a doubt, we do have to work on that language, and that's an important part of this. Yeah.
I'm very curious about your recruiting practices and what you do and what you're on. If you could give me a super high level of how you do recruit and how much budget you put towards it. This is the marketing person here.
Yeah.
And just in addition, just a curious question, have you heard from your users, or do you have any belief that an app would change things versus being web-based?
Yeah, great question. So let's start with the app thing. I wanted to maintain one platform as well as I could, and at the time, we chose the web application. Do I think some people would like an app? Absolutely. And I think it would be more powerful to offer both. So choose your own adventure type of thing would be really good. But there's also a group of people who say, I don't want another app on my phone. So, it's that balance. I think if you can offer both, I would go that direction. Recruiting. So, yeah, I could do a whole hour on recruitment approach. approaches. So number one, you have to be present and there every day. So we run a Facebook page in English and a Facebook page in Spanish. We have an incredible social media team that runs that, responds to every comment they possibly can. We advertise, we do paid advertising in Facebook, in X, in Instagram. TapJoy is also paid advertising. So budget. Believe it or not, we only spend about $2 per participant that comes in the door. So it's really incredibly cost effective. But I told you we see 700 new participants a day, so our daily budget is quite large. So that's one of the other things that folks need to think about when you're on the internet. You could be a victim of your own success, right? You could wake up tomorrow with a pretty big bill because your ads went viral. So it's like, you know, it's a tough thing to balance, but we've learned over time. So I don't know if that's super helpful or not, but I think the main take home for you though, and you know this because you're the expert, is that scientists oftentimes think they can do everything because, you know, they've been trained to think out of the box and teach themselves, right? That's why experts exist. So you can't teach yourself how to be an expert marketer or a social media person. If I had tried to run this myself, thinking I could do it all myself, it would have failed miserably, the social media side. And so I think it's really important to know when you need to engage with experts. And you run into that more often as you start to do things which are a little more out of the typical space, like living on the internet or living in a mobile lab. Yeah.
Sandy?
Excellent talk. Thank you so much. I wanted to zoom in a little bit to the actual research part of it. How do you find your data? What do you test? What does a memory test look like? Can you walk me through that part?
Yeah, sure. So let's see. So the 10-minute MindCrowd experience, we collect demographic information, about 30 questions. It's exactly what you're imagining. And then the two cognitive tests are as follows. One, there's a processing speed test, which is basically simple visual reaction time. So a pink ball appears on the screen, and all you have to do is react to it as quickly as you possibly can. That's a measurement of your processing speed, your brain's processing speed. That test takes about 1 minute, 2 minutes maybe. Then the memory test takes about 5 minutes. So it's paired associate learning. So you see 12 word pairs, one after another, and you only get to see them for 2 seconds. So it'll show you 2 words, and then another 2 words, then another 2 words, go through all 12. And then we present you with half of the word pair, one of the words, and you have to type in the second word. After you do that for all 12, you go into round 2 and you get to see the correct word pairs again. And then we ask you to type them in, and then you see it for a 3rd time. So people, the score for each round tends to improve because you start to capture additional word pairs with each round. Yeah. Was that what you were hoping? Okay. And we have a lot of other memory tests, and executive functioning tests, and other types of cognitive tests that I just couldn't cover today. But that's part of our MindCrowd VIP experience, and you can join us and participate.
Hi. I think it's wonderful that there's so much research being done, but are there physicians who want to work with people who want to know where they are and what they can do?
Right. So that is an area of interest that we're working on right now. And that gets back to my slide, when was your brain last tested, right?
Yeah.
We want that to be tested in a medically meaningful way where your doctor can help interpret the results and see how you change across time. So that is a grand vision, but we are working on that. And so maybe as an example, we could look at the tests that physicians administer today that are memory tests, right? This is something like the MMSE or the MOGA. These are really designed to detect frank disease, overt dementia. And so they're not very sensitive just to normal aging because just the way they're designed. But they're also built on a normative data set. So that's part of the equation as well, is to have this large normative data set like we have to provide some context for the results.
Yeah, but I'm thinking about just neurologists who would want to work with somebody. I think I once asked someone here who was a doctor, and she said, "Oh no, don't go to a neurologist. They just want to know what's wrong with you." I mean, are there neurologists who would want to work with somebody who wanted to have MRIs and know where they were and so forth?
I think there absolutely are. It's a great question, but it opens up the whole can of worms about how much time can my physician spend with me. And I think we make a bigger impact if we're not with the neurologist, we're actually in the primary care physicians, where this becomes part, you get your cholesterol test, you get your cancer screening ordered, and then as you're walking out the door, they say, by the way, we also want you to do this test. You can do it at home, you can do it on your smartphone. And so getting into that primary care, wellness visit area is where I think we make a bigger impact.
Just a quick question, and then I'll hand it to Sandy. I'm curious because so much of our work here focuses on longitudinal engagement. And just given the breadth of reach that you've had, what do you think is the feasibility of using some of these same recruitment approaches or whatnot to facilitate more longitudinal continuing engagement? Because that's been a challenge.
Yeah, no, it's always a challenge, but I do think that some of the social-based approaches are really powerful for that. One of the things that we really have found is helpful for longitudinal engagement with the mobile lab, we actually 3D print your brain using your own data, and it shows up at your house like a month or two later. And it really helps remind people, oh yeah, I did this cool science study, now my brain's in the mail. So you have to be super creative to keep your folks engaged. And we do have a longitudinal aspect of our study that's ongoing. I just didn't focus on that for today, but we've had about 10,000 longitudinal participants. But it is really a struggle. You see the numbers really dwindle, especially if you don't put a lot of effort into it. So I don't have a magic answer for you other than it's hard.
Matt, congratulations on a great talk and work and your imagination and innovation. I'm so glad that we're here together and can find ways, because you're talking our language in terms of what happens. My question to you is back about the ApoE4.
Yes.
Because we know that, I didn't see the gender difference, that women are when they have two, it's much higher risk, maybe even as much as 40 to 50% to have Alzheimer's than a male. Did you look at the gender difference? And then did you look at how many people have it that don't have memory problems? Because that's the story we need to tell.
Right.
Because everything's always about bad if you have both. But if you could say, these people had ApoE2 and they're above ApoE4, two of them, and they're high performers. That's the story no one tells.
Yeah, for sure. So we haven't yet looked at some of the interactions. So for example, is sex an interaction term with the ApoE carrier or genotype status? That's definitely next on our list, for sure. And yeah, I mean, I think there's a whole story to tell here about how, look, I'm a geneticist, so I wish genetics was your destiny, and I could just tell you flat out by doing a genetics test, right? No, of course, I'm being silly, but it's not, right? I mean, that's the whole point, is that it bends these curves a little bit, but it's not the be-all, end-all, and we're all thankful for that, because we can't control our genetics. We can control how we, how we approach our life, how we do brain training in our environment.
But the story's not out there, because the risk is always, and I love what Steve's question is, it doesn't mean, it's a risk, but it doesn't mean that you're going to go there. And I think we just need to tell the other side of the story.
For sure, agreed.
Okay, I have our question from the internet. Oh, sorry.
Okay, no, okay. So in your study looking at the aging process and you got that menopausal bump, did you look into hormone replacement therapy as a cause?
Yeah, so we have a whole women's health survey that we've administered to about 6,000 participants. And it seems like the answer is those women who did not do anything related to hormone replacement or surgery perform, those women who did not do any of those things are the higher performers within the women group. Yeah, so that was a lot of gasping. Yeah, I know. So the data are what we see, and it tends to be a little different than what's been out there published already. However, I feel good about the numbers that we have in the data. So yeah, that story is still developing, but that's our current finding.
All right, and so now, this is our last question. It's from online. Do you have any dream biomarkers or task-based tests across any of your channels to inform your research? And if anything, do you feel that there's a gap? Yeah. I mean, dream-based biomarkers, meaning biomarkers of dreams, or man, both of those sound really cool. I think we all want to have some clarity about our future. We want to know about our trajectory because I think it's a huge motivator to try and change that. And so biomarkers that would be leading biomarkers, not lagging biomarkers, things that would help predict a little bit about where we're going would be the dream. Do I think there are gaps? There are a lot of gaps. And it goes back to the first slide I showed you guys with the wrecked car. We have incredible biomarkers now for Alzheimer's disease dementia. They started in the brain, then they went to the CSF. Now we have great blood-based biomarkers to help clarify your risk or a current diagnosis of Alzheimer's disease dementia. We don't know, and we don't have a great collection of biomarkers that translate well to the healthy, normal aging brain that predicts cognitive performance in the future. And it's not clear if those biomarkers about dementia are going to be useful to predict your cognitive trajectory, or if they're only sensitive to disease. So I think that's the area where I want us to get better at. Yeah. Perfect. Lifelong brain health, that's our shared mission. Thank you so much, Dr. Huentelman, for coming and sharing all of this fascinating data with us. We're looking forward to incredible collaborations, and we just appreciate you coming and sharing this. Thank you, everyone, for coming and joining us today, and we look forward to seeing you again on March the 21st when Dr. Stephen Lombard will be our guest speaker. Dr. Lombard's lab at UT Arlington has pioneered the use of focal cooling to reversibly deactivate regions of the cerebrum, examining cortical plasticity in the presence and absence of acoustic input, and following the initiation of auditory processing through cochlear prosthetics. Before we wrap up, I want to encourage everyone to pull out their cell phone and focus on this QR code, because we would love for you to join us in this new sleep tech challenge that's so cool, so much fun, and such a good thing. Thank you.